Hi everyone, I'm Dr. Redfield. Welcome to the very first lecture of the first module of Useful Genetics. This lecture has an abbreviated version of the structure that you'll see in the whole module, in that we'll start and end by thinking about human genetic differences, and in the middle we're going to introduce some very basic concepts, in this case just the very first introduction to DNA. Now, as people, we're really good at noticing differences in appearance of other people. That's probably because we're social animals. We're also really good at noticing the ways in which people from any particular part of the world look different from us, but very much like each other. And it's often said, well, we're all the same under the skin, but are we really? This is a question that genetics can answer. To think about this, we need to introduce two very important terms that will come up again and again and again, and that's phenotype and genotype. These terms were invented by the very first geneticists as a way of distinguishing between two things. First, the phenotype was the observable properties of organisms, what you could see, the features that we cared about. And it's come to include not just what you can see with your eyes, but the observable, the molecular properties, behavioral properties, anything about an organism. When the term genotype was invented, it was because of the brave inference that these phenotypic differences that we see were caused by differences in some mysterious entities which were given the name genes. Different versions of genes were the different genotypes. We now know that this was a correct inference, and in fact, that the different versions of genes are in fact different versions of DNA sequences of chromosomes. Now, quick refresher of information about DNA. All of our genetic material is organized into long molecules called chromosomes. And these long molecules are chains of subunits called bases. So it's a long chain. And in the DNA and other what are called informational molecules, the bases aren't all the same. Instead, they come in four slightly different versions, four slightly different subunits, called by the names A, G, T, and C, which are just the single letter abbreviations of their chemical names. We often represent DNA as the order of the bases in the sequence because, in fact, this order of the sequences is the genetic information. It is the differences that cause the phenotypic differences that we see. So, if we think about DNA in the life cycle, we know that each egg and each sperm contains one complete set of DNA molecules, 23 chromosomes. And when the egg and the sperm meet, when the sperm fuses with the egg, the resulting fertilized egg contains two complete sets of genes, one from each parent. Individuals develop because the fertilized egg undergoes many, many cycles of growth and cell division, producing all of the cells in our body. And most importantly, from a genetic perspective, all the cells in our body, for instance, every cell in our arm, all our cells, contain the same two sets of genes that were in the original egg and sperm. Now, what is this complete set of DNA molecules? Well, as I said, it's 23 long chains called chromosomes. The other very important information is, well, how many subunits are we talking about? We're talking about 3 billion subunits, 3 followed by 9 zeros. Now, we speak of this complete set of DNA molecules as being a genome. And we can use the word genome in two different senses. The first is we can speak of the human genome, by which we would mean a reference sequence, like might be stored in a computer. But really, when we think about humans, we need to think about the genomes of all humans and the genetic diversity between us. Together, this makes up the real genetic endowment of all humans. Now, how different then are our DNA sequences? Well, from one perspective, they're very, very similar. So on average, they're 99.9% identical. A way to visualize that is to consider the set of DNA that you inherited from your mom and the set that you inherited from your dad, shown here in blue. Here I'm just showing 1,000 bases. If you compare them, what you'll find is that on average, in every 1,000 bases, there's just one sequence difference. Now, what if we were to compare a set of DNA from you and from one of your neighbors? Again, we pick 1,000 bases and compare you and your neighbor. And what we'll find is, well, on average, there's one DNA sequence difference. Even if we were to compare two sets from people from other sides of the world, again, we'll see on average there's just one difference. So it doesn't make any difference really how far away we come from. We're all very similar. But as I said, there's two perspectives. And the other perspective is to think about, well, how different are we? So I'm going to let you calculate this in the first of what are many self-test questions. And the format will always be, there's a slide here where I'm introducing the question to you, asking you to calculate how many genetic differences are there between you and your neighbor. And on the next slide, you'll see Coursera's version of this question. And after you've done the arithmetic, you can check your answer. And then finally, we'll come back to a slide where I'll discuss the answer. Here's the Coursera question. I hope you got that right. The right answer is there's 6 million differences, a lot of differences between you and your neighbor, between each of us and each other one of us. Where did this number come from? Here's the calculation. There's one difference per 1,000 bases. There are 3 million bases per set. And there's 2 sets per person. So that gives us 6 million differences. So how much difference do these differences make? Well, that's kind of a silly question. If we put it genetically, we will ask how much phenotypic difference do these genotypic differences make? And the answer is, well, most of these differences don't make any detectable difference to phenotype. And that's because most of these differences are in parts of the genome that are really not important for determining our phenotypic properties at all. We'll talk about this a lot more in a later lecture. But the rest of the differences cause all of the heritable differences between people. And just in case you're thinking that people are kind of special, we're not. Exactly the same situation exists for all of the other organisms on the planet. Their sequences of any one species are very similar, but there are so many bases that there's still a lot of differences which cause all the phenotypic differences. Now, what we've done, we've introduced some new words, especially phenotype, genotype, and genome. We've talked about the genome in the life cycle, and we've talked about the differences between the genomes of different people, both how similar we are and how very, very many differences there are. Now, coming up next, we're going to talk in more depth about the properties of DNA. And following that, we've got a series more lectures in Module 1 talking about the properties of genes, of chromosomes, of life cycles, and finally, how we can compare DNA sequences to learn about human history. I hope to see you there.